Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 7th July 2020. In this we are going to study about secondary growth. Secondary growth involves formation of secondary tissues, and the secondary tissues are formed by two types of lateral meristems. We were talking about the meristems, meristematic tissue, tip apical meristematic tissue and lateral meristematic tissue. So the lateral meristematic tissue being present on the lateral sides, means on the sides, is responsible for secondary growth. So you can guess that this adds to the girth or the width of the plant, not the length. Hence, this secondary growth involves mainly increasing the bulk of the plant, either in the stem or root. So secondary growth, basically it is cambium, vascular cambium. So this vascular cambium and cork cambium, two tissues are mainly responsible for formation of this. Vascular cambium is formed from conjunctive parenchyma and pericycle. There are two tissues which form vascular cambium. What are those? Conjunctive parenchyma and pericycle. And the cork cambium, this is also called phalangen, and the phalangen is formed from pericycle. And cork cambium produces phellum. Cork cambium produces phellum, cork cells on the outer side, and phelloderm or secondary cortex on the inner side. Phellum and phalangen and phelloderm together constitute what is called periderm. So periderm is combination of phellum, phalangen and phelloderm. There are certain loosely arranged areas in the periderm formed due to rapid activity of phalangen. They are called lenticels. Lenticels help in gaseous exchange of transpiration. And coming to this side, vascular cambium cells are of two types, as you are saying. One is fusiform initials, they are vertically oriented, and ray initials, horizontally oriented. So fusiform initials and ray initials. And fusiform initials give rise to very important secondary xylem and secondary phloem. That will add girth to vascular bundles. And ray initials, which are horizontally oriented, will give rise to medullary rays in the secondary tissue, that is vascular rays, xylem rays and phloem rays. The activity of cambium is not uniform throughout the year. Secondary xylem formed during spring has wider elements, and that formed during autumn has narrow elements. So due to differential activity of xylem, annual rings are formed consisting of spring or early wood or autumn or late wood. With the time, the tracheids and vessels of inner part of the wood get plugged by ingrowth of adjacent parenchymatous cells into their cavities through the pits. This wood becomes non-functional and is called heartwood. So the outer functional wood is called sapwood. Now let's see, the secondary growth is mainly, as I said, is responsible for width, girth of the plant, and it is because of the lateral meristem. So it is mainly responsible for increasing the girth. Secondary growth in plants are produced by lateral meristem, and instead of growing in length, as I said, secondary growth is radially increasing. That means it is adding to the girth. And secondary growth arises in regions of woody plants where primary growth has ceased. And what is important is secondary growth starts only when undifferentiated cells are formed. Now look at this point. The process starts when differentiated cells revert to become undifferentiated cells, forming two meristems called vascular cambium and cork cambium. So look at this here. Primary growth in stem, it will be like this. Focus on primary xylem and primary phloem. And in secondary growth, look at that. we along with the primary xylem and primary phloem, we have got secondary xylem which is forming a ring, and secondary phloem again a ring. Now look at the radius or the circumference of the stem when we took the section. In primary growth, what is the circumference? And in secondary growth, what is the circumference? Naturally, many rings have been added, and that is the purpose of secondary growth. So vascular cambium produces vascular tissues which are called secondary xylem and secondary phloem. And cork cambium, pelagian, forms initially parenchyma cells. And the secondary growth in dicots and conifer stems begins when a vascular cambium cells arise from residual procambium. This is important. And vascular cambium cells are existing cells that becomes meristematic at different times under the influence of hormone auxin. Auxins are responsible for this. And look at this in a stem, vascular cambium formation in a stem. You can see the ring, initial formation also. Compare with the growth after several years. Look at the girth first and look at the rings that have been formed. Secondary phloem, look at that. Secondary xylem, look at that. Pelagium, cork cambium. So many rings have been added. And the vascular cambium is mainly for secondary xylem and secondary phloem. And look at this root, vascular cambium formation in a root. In root also you see initial formation. You got primary phloem alternating with primary xylem. And look, the girth is very small. And in growth after several years, look at that. Primary phloem and secondary phloem. Secondary phloem almost formed a ring. And look at the pericycle. And secondary xylem has formed one more ring. So you got a series of layers being formed, and pericycle got enlarged. And look at the most recent phallodome is outside, and old one is inside. And previous layers also you can see. So overall, secondary growth is meant for increasing the girth of root as well as stem. And this forms what we call secondary xylem, is what we commonly called wood formation takes place. And further, the secondary growth maintain two basic functions of vascular tissue. What is that? Conduction, that is transportation, and support. There are basically two types of dermal tissue in vascular band: epidermis and peridermis. And the epidermis and cortex form during primary growth, and periderm will produce cork cambium. And cork cambium consists of cork, phallodome, and cork cambial cells. Now, cork itself scientifically is phellum. It forms outside of the cork. And phallodome is thinner layer of living parenchyma cells. Unlike vascular cambium, cork cambium does not grow in diameter. And let me show you some pictures in this. Now, bark. The bark is the part of the stem or root surrounding the wood. Bark has two distinct inner and outer bark. Okay, and the rough texture of the bark is due to splitting of older layers of periderm. And now, two types of meristematic cells that form vascular cambium: fusiform initials and ray initials. These initials undergo periclinal and anticlinal cell division. Of course, that is due to cell division. Now look at this. This is how the vascular cambium and the surface of the root. And you can see the anticlinal cell division and periclinal cell division. Both the sides increasing the girth. And here you can see the periclinal and anticlinal division further. And look at the width that is getting increased. And the fusiform initials will be like this. That is like a spindle it will be. And these divides, one daughter cell becomes either xylem or phloem. These are responsible for conduction. And vascular cambium over time, look at that, how it is helping to grow further. And fusiform initials also responsible for conduction of water. And ray initials, horizontally arranged cells in the vascular cambium, as I said in the beginning, it helps in producing flow rays and xylem rays. Now look at this three-dimensional vascular tissue in a stem. This is a primary xylem, secondary xylem, primary phloem, and look at the ray initials, just like building and fusiform initials. So this is how the secondary growth occurs, and the apical meristem is responsible for the fallout. And finally, what we need to focus is secondary tissue, in which bark and wood, and the bark and wood are very wood is the secondary xylem of the seed plants. And look at this is what I want you to focus on: sapwood and heartwood. Now you can see the bark, first layer bark, then second layer secondary phloem, third layer vascular cambium, then heartwood, and centrally there is a sapwood. And sapwood is, as you are seeing here, is the outer wood of the old stem, and heartwood is the central wood of a old stem. It is light colored. Sapwood is light colored and heartwood is dark colored, as you are seeing that. And leaving cells are present in sapwood and leaving cells are absent in that. Sapwood is functional part of the secondary xylem of the wood. Heartwood is the non-functional part of secondary xylem. And sapwood is lighter, heartwood is heavier. And sapwood is less durable because it is susceptible to attack by pathogens and insects. And heartwood is more durable due to its little susceptibility to attack of the pathogens. This is how we differentiate heartwood and sapwood.